Welcome back to my course Aspects of Biochemical Engineering, and we are in the last class I was discussing of activated sludge process. Now, if you look at the biological process that where cells, living cells are growing, they usually grow in two different mode. One is aerobic, another is anaerobic mode. The aerobic last activated sludge process, basically it is the aerobic fermentation process, and now I shall discuss the anaerobic fermentation process. Now, I told you that this activated sludge process is used for wastewater treatment purpose. Now, any industry that whenever they submit any kind of proposal, but they have to show that that whatever waste material they are generating, that should be properly treated, so that the environment should not be polluted. So, we have found out that 10% of the total investment of an industry that usually go for the wastewater treatment purpose. Now, if you look at the history of wastewater treatment process, that most of the industry, when they invest the money for wastewater treatment process, they consider there is no return from that wastewater treatment process. Whatever money is spent with that process, that will go in waste. But due to the invention, due to this anaerobic digestion process, now it is possible to get some revenue from this particular process, some money we can earn from this particular process. So, this is a very interesting process through which the lot of industry they are operating the process, and I want to mention here that in the Western country, particularly if we visit Germany and Sweden, you will find that some buses they are operating by using the biogas, and biogas is nothing but it mostly contains methane and carbon dioxide, and carbon dioxide you can easily remove through the absorption process, and then this methane we can use for the operation of vehicles for as a particularly I can tell you in the distillery industry, they required lot of heat energy for the distillation of alcohol, and it has been found that if by using this anaerobic digestion process, the 50% energy that is required for the distillation process, that can be met from this from the wastewater treatment by anaerobic digestion process. So, with this small introduction, let me go to the anaerobic digestion process. The anaerobic digestion process, if you look at it is comprises of series of biological process in which the microorganism break down the biodegradable material in absence of oxygen. So, you know that this is anaerobic fermentation means in absence of oxygen. Only I want to highlight here that organism when grow aerobically, they require their amount of cell mass formation will be very high, but when they grow under anaerobic condition, amount of cell mass formation will be very less. So, so in other way, indirectly I can tell you that the nutrient requirement for the anaerobic fermentation process will be much less as compared to aerobic process, because since aerobic process your cell mass growth is about 10 times as compared to anaerobic process. So, your nutrient requirement for the aerobic fermentation process much high as compared to anaerobic fermentation process. So, now here all reactions is occur in absence of oxygen, and this is one of the oldest fermentation process. This is the oldest fermentation process that stabilize the sludge obtained by activated sludge process. Now, I was discussing in the last lecture that activated sludge process. Now, we should remember that I told you that when you talk about this soluble organics, we are converting to cell mass and carbon dioxide. The carbon dioxide that is going out of the system, but what is the cell mass that remain in the system, that we cannot throw it out, because it contains lot of nutrient. As soon as you throw it out, then all the bacteria, other bacteria present in the atmosphere, they will try to grow on this solid material and degrade the material. So, your environment will be polluted. So, this sludge we can do to be further treated. Now, this sludge either you pass through the incineration process. So, you can produce lot of heat. The problem is that it contains a very good amount of moisture. So, you have to remove the moisture before you do you burn this material. But best way of treating this material if you pass it through the anaerobic digestion process, so that this organic material can be converted to methane and carbon dioxide. So, this is exactly what we have mentioned here. This is the oldest process used for stabilization of the sludge obtained from the activated sludge process. This process capable of trace solids as well as the liquid waste. This is widely as a source of renewable energy. The process produces a biogas consisting of methane and carbon dioxide and other trace gases. The trace gases means, since the anaerobic, we know the anaerobic fermentation process, that dissolved oxygen concentration of the media is not required, is absence of molecular oxygen. Though we know for the respiration of the microorganism, respiration of the living cells, always they require the oxygen. Now, question comes, from where they get the oxygens? They get the oxygen from the compound like sulfate, nitrate, nitrite. The sulfate, when they use as a source of nitrogen, then sulfate will be converted to sulfide and ultimately is converted to sulfide and H2S. So, that H2S gas that is produced in this system. So, one of the problem that we have with this biogas generation process is the H2S present in the gas, though it is very trace amount, but may causes the may affect the material of construction of the pipeline. Mainly two groups of microflora are present. One you call this is bacteria and archaea, this is the present, and now, let us see that what are the major steps involved in this anaerobic digestion process. The first, what we have, the complex organic material here. This is the influent, and then it comprises of what polymeric material. This is protein, is the polymer of amino acid, carbohydrate, this is polymer of glucose, and maybe monomer, dimer, pentose, hemicellulose, cellulose might be there. Then lipid, we have the esters of fatty acid and alcohol, and this when hydrolyzed, it produces the amino acid and sugar, that is soluble. What do you call, this is the insoluble material, and when you convert it to the amino acid or sugar, we call it solubilization of this process, and this process we call hydrolysis. And this hydrolyzed material then converted to the different volatile fatty acid like acetic acid, butyric acid, lactic acid. So, different type of acid formation is there, and this higher acid again converted to acetic acid before it produces methane and carbon dioxide. And in this process, this is acid generation process, produce hydrogen also, and this in combination produce the methane and carbon dioxide, and finally we call it biogas. So, basically if you look at this process, it is 4 major steps are involved. One is called hydrolysis process. Hydrolysis is basically liquefaction process, and the acidogenesis process, that means during the acidogenesis process, this different organic acid, volatile fatty acid formation take place like butyric acid, acetic acid, lactic acid, fumaric acid. The different type of acid formation is there, propionic acid formation is there, and this higher acid they convert it to methane via acetic acid formation, what we call acidogenesis. And this acid, when acetic acid, we know what is the formula of acetic acid, CH3COOH. This is converted to methane and carbon dioxide. So, this is how it is produced. Now, classification of anaerobic digestion technologies, this can be, we can classify it at the high rate and low rate. High rate, this that hydraulic retention time is less than a solid retention time, and the treatment of the wastewater, this is the low solid content, the total solid is 4 to 10% is used. use the biogas settler, anaerobic baffle reactor, anaerobic filter, upflow anaerobic sludge bucket reactor. These are the different reactors used. But in case of low rate reactor, where no mass retention is there, the hydraulic retention time equal to solid retention time. So here the treatment is slurry or manure or wastewater. Mainly the solid material we use. This is contains the two type of solid. One case the total solid concentration less than 20%, another is the more than 20%. So different type of anaerobic biogas digester that is used here. Batch and fed batch process, plug flow reactor, continuous start tank reactor is used, and here in more than 20% you have anaerobic biogas digester, batch process, plug flow reactor, continuous start tank reactor is used. This is the how we can classify this anaerobic digester process. Now we have different anaerobic digesters commercially available throughout the world, and this is the upflow anaerobic sludge bucket reactor. Upflow sludge bucket reactor appears to be the very efficient reactor. Now what is that reactor? This is the reactor is like this, and so here we have some kind of sludge that here we have this. So this is we can take out the gas, we can take out the liquid from here. So this is the liquid we had. So here sludge formation is there. This is feed we have, and this is the gas will coming out from the top. And due to the high concentration of cell mass here, when the feed is going like this, then it converted to immediately methane and carbon dioxide. And main purpose is that this conical the things, particle, they will strike one another, and ultimately it will strike here. It will come in this direction. Again, one particle strike this, then ultimately and come in the, and since it is coming in the bottom direction, then all the sludge slowly this accumulated at the bottom of the reactor, and time to time you have to drain out the sludge, because we have to find out what is the optimum height of the sludge for getting the maximum rate of reaction. So this is largely used. This is how it looks. Then covered anaerobic lagoon digester. This is how it is operated. Plug flow reactor. Here there complete mixed digester. This is there, and dry digester system and fluidized bed reactor is there. So these are the different. They are designed based on the organic waste stream trends, such as the manure, municipal wastewater treatment, industrial wastewater treatment, municipal solid waste. Some commercially available digesters are included. Now applications of anaerobic digester, there are several. This is the input. Input is maybe energy crops, maybe manure, maybe industrial waste, maybe the source separated, the organics or municipal solid waste. The restaurant, we have food industry, food waste, the wastewater treatment from the plant sludge. This pass through the anaerobic digestion process, and then whatever methane is produced, that can be used for the generation of electricity. And we have biogas, we can use as running the vehicles, we can use as a renewable fuel. If the digested material can be used as a biofertilizer, and reusable, usable for water and compost. This is largely used for composting purpose. Now, if you look at the digester, you have several sections in the digester. This is this portion is very interesting. The reason is that in this portion, that active digested sludge, the most of the organisms, they are active in this particular portion. Now here the digested material that we had. Now what we have observed, we have several anaerobic digester plants located throughout the world, and when big digester we operated, major problem is that long time operation, maybe after several months of operation, they will find that this digested material, that accumulates at the bottom of the reactor, and there they settle in a very thick sludge and very difficult to separate it out. So a time will come, this sludge will keep on rising, and if it rise 50%, then you have to stop the operation, and then we shall have to take the material out to regenerate the process again. So, this is very important, this is supernatant layer that we have, and gas is removed like this. Sometimes we have here some kind of crust formation is there, and if we use some kind of agitation, or we scrap the surface, then gas will escape out. We can increase the gas production. This material can be used as the digested material, is as the biofertilizer. Now, process design that includes that the liquefaction I already explained, acidification also explained. Acidification I also explained that the higher organic molecules like butyric acid, propionic acid, they convert to acetic acid first, then carbon dioxide, as well as carbon dioxide and hydrogen, then to methane and carbon dioxide. And methanation, mostly the acetic acid or methanol, they convert it to, or carbon dioxide and hydrogen, they convert to methane. So, in case of liquid waste, last 3 steps are important, because in case of solid waste, all the 4 steps are plays very important role. Now, if you look at this, this is very important, the process design of this process, that concept of mean cell residence time is used to describe the process design of, which we already explained that what is called mean cell residence time, and design is similar to the activated sludge process. Respiration, oxidation, end product of anaerobic digestion are methane and carbon dioxide. The equation is V into this. This is very important. Now, let me explain what is meant by that. Now, Q0 or Q or Q0 is same, that is incoming flow rate of the digester, because suppose let us assume this is the digester and this is Q0, am I right, or Q, and this is the S0 is the, so what is the input amount of substrate input in the system, Q0 into S0, am I right, and then E is the efficiency of the waste utilization, normally range from 0.6 to 0.9%. So, and the question comes, what is the percentage of this organic matter that can be converted, they can be utilized, 60% to 90%. So, E is that factor, and then I told you when it converted, a part also converted to the cell mass, and that remain in the system. So, that is to be deducted, am I right? If you deduct that, then this is the actual the BOD that is removed. Now, if you multiply it by 0.35, you will get the volume of methane produced. Now, question comes how that figure has come. Let me explain that. Now, let us assume this here that what is that 1 mole of glucose, 1 mole of glucose produce 3 moles of carbon dioxide and 3 moles of methane. So, this is 180 and this is 48, because 3 into 16 is 48, am I right? And so now if you convert it into BOD, the 3 moles of methane require 6 moles of oxygen to convert it carbon dioxide and water. So, if you have the ratio that kg methane produced per kg of BOD, how you can write the 48 gram methane you can produce for 180 grams of glucose, and 190 grams of oxygen required for the oxidation of this methane, that is 192 divided by 180. That means the thing is coming that 0.25 kg of methane produced per kg U of BOD removed, am I right? Now, we know at NTP 1 mole 1 gram mole of the gas is equivalent to 22.4 liters, am I right? Now, if you put that equation here, then we will find this will be equal to 0.35 cubic meter per methane, and cubic meter methane. So, what I want to tell, now in this equation you see that this is the amount of BOD removed. So, we know this is per kg of BOD removed. So, you can easily find out how much volume of methane is produced, 0.35 you can find out, and if you multiply it by 0.25, then you can get the kg of methane produced per kg of BOD removal. So, this is how you can calculate that. Now, in the process design, another parameter very important, that what is the biological solids, because if you look at the mass of biological solid synthesized daily, that is rate of cell mass formation. How you can calculate that? The Q into S0, because I told you that you can remember that Q0 into S0 into substrate conversion, am I right? And if you multiply this, that is the actual amount of substrate that is converted. Now, if you multiply by Y observed, x by S observed, then what will happen? You will get what is the exact amount of cell mass that is produced. This is exactly, that is this is capital Y, this is capital Y, the x by S, that is Y stands for, that is the yield coefficient. Okay. Now, this is the typical different kinetic constants that we have as far as anaerobic digestion is concerned. The domestic sludge, that yield coefficient, and mu d is the specific death rate of the cells, that varies from this range, and typical value is this. Okay. In case of domestic sludge, in fatty acid we have this this typical value is like this, and carbohydrate it is 0.024 and 0.030, and proteins is 0.075 and 0.016. Now, if you consider the mean cell residence time using the design continuous start tank reactor, then we find that 8 degrees centigrade, the theta c value, that is the mean cell residence time, will be 11, but the suggested is 28 days. Now, in case of 24, it is 8 and this is 20. In case of that 30 degrees centigrade, it is 6 and it is 14. So, what I want to mean, as you increase the temperature, your kinetics is better, because your rate of conversion of the gas will be more high, it will be higher as compared to low temperature. Your organism will be activated, because that we find that reaction kinetics is parameters improve as you increase the temperature, because we know Arrhenius equation, that how the rate constant depends on the temperature. So, this is this we observe in case of particular anaerobic digestion process, temperature plays very vital role. And here I want to tell one particular matter, that that you might be knowing that that in India we have millions of biogas plant that is located in different rural areas, and also China, also they have millions of biogas plant in the rural area, because if you look at the social structure of China and India is more or less same, and because our standard of living is not as good as the Western country, and that is why this process largely implemented in India and as well as China. Now, what we observe that that in case of China, that this process is quite successful as compared to India. The reason is that in the China they have certain analytical devices, that how this small this biogas plant can be operated. They have the monitoring system of pH. Now, I can tell you the anaerobic digester usually governed by two type of organism. One is the acidogens and methanogens. When both the organism present in the reactor, so both acid conversion and acid to methane conversion take place. Now, if there is if the rate of acid formation is high as compared to rate of methane generation, then what will happen? The acid concentration in the reactor will increase. Now, as the acid concentration increases, the pH of the reactor will go down. As the pH goes down, the rate of methane formation will be reduced significantly. So, this is the major problem. So, those things you have to take care. Another thing is that the temperature, because we know that though we are in the tropical country, but during winter our temperature reduces as low as 15 to 20 degrees centigrade. So, naturally our temperature of the digester, if we do not have any kind of heating system, and though we know that the biochemical reaction is exothermic in nature, but the temperature will go down, am I right? As the temperature goes down, then your rate of reaction also decreased significantly. That is the major problem that we have with the anaerobic digestion process. So, temperature is a very important criteria that we shall have to maintain. Now, this is the biomethanation pilot plant that I personally work at IIT Delhi, and here we use the distillery effluent. This is the acidogenic reactor, acidogens genic reactor, and this is methanogens genic reactor. Okay, and this is the feed tank that we have. So, what we do, we can put the distillery effluent here, and then we can continuously pass through this, and then after that is you can easily find out the size of the acidogenic reactor is much small as compared to methanogenic reactor. The reason is that acidogenic organisms, they work very fast as compared to methanogenic. Methanogenic reactor is a very slow growing organism as compared to acidogenic organism. So, here the reaction, since it is very fast, the rate of acid formation will be very fast, is required very small reactor, and here methanogenic reactor is very time consuming, slow growing, so we have to keep the bigger reactor. So, this is how we operated this reactor. Now, we have taken one problem that they estimate the size of the digester required to treat the sludge from a preliminary treatment plant design 38,000 cubic meter per day of wastewater. Find out the volume loading and they estimate the percentage stabilization and amount of gas produced. From the wastewater to be treated, it has been found that the quantity of dry solid and BOD removal is 0.15 kg per cubic meter and 0.14 kg per cubic meter respectively. Assume the sludge contain 95% moisture and has specific gravity of 1.02. Other pertinent design parameters is given. Hydraulic system of the reactor is continuous flow start tank, and solid retention time is 10 days, the E is 0.8, and waste contain adequate nitrogen and phosphorus for biological growth, and Y x by S is 0.05, mu D is 0.03 the inverse, and constant are all constant as temperature as 35 degrees centigrade. So, this is the problem that we have. Let us see how we can solve this problem. Now, first we shall have to find out the sludge volume, am I right? Now, how you can find out the sludge volume? So, this is the what is the content, the dry solid, this is the 15 kg per cubic meter, am I right? So, 15 kg per cubic meter, and this is the volume of that liquid is coming in. So, if you multiply that, you will get the amount of sludge that is entering into the system. The density of the sludge is 1.02. So, if you multiply it by factor, and so you will get this and another factor that is given, the 95% moisture content, then 5% is the total solid, am I right? So, if it is like this, then we can find out the volume of the sludge is 111.8 cubic meter per day. Now, BOD loading, we can calculate, this is the BOD 0.414 kg per cubic meter. This is the amount of this liquid that is entering per kg, that is this is per day. This has some mistake is there. So, you correct it, and this you can find out, this is 5,320 kg per day. Now, digester volume, how you can calculate? This is Q. Q is this is volume of the digester, and the solid retention time is the 10 days. So, if you multiply it by this, that we can find out the volume of the reactor. So, and then volumetric loading, how you can calculate? This is the amount of sludge that is coming in, and this is the volume of the reactor. So, amount of solid material that is putting per cubic meter of the reactor per day is equal to that you can calculate very easily. Now, if you want to find out the cell mass formation, this is the equation that we have, and if we have all the values in this problem, if we put that, we find it is 163.7 kg per day. Now, we have the percentage stabilization. How you can calculate that? This is the BUT that is removed, and this is the cell mass, that is the BUT equivalent to the cell mass. So, actual removal of the BUT is this, and this is the Q into S0 is the total BUT that is input in the system. So, if you do that, you can easily get the percentage stabilization of the solid material, that you can easily do that. And then finally, we want to find out that what is the amount of gas produced. So, we have already developed this equation, that 0.35 into EQ is 0, 1.42 into Px, all the parameters is already estimated. So, we can find out 1408 cubic meter methane produced per day. Now, if the methane contains, if the gas contains 67% of methane, then we can find out the total gas produced about 2102 cubic meter per day. So, we can estimate this very easily. So, so in this particular lecture, I try to discuss the anaerobic digestion process. The anaerobic digestion process is considered the most useful process, because here we can generate lot of methane, and this methane can be generated both from the solid as well as from the liquid waste. And this I told you, this process has 4 different steps. One is called hydrolysis, then acidification, then acidification, then methanation. This is usually governed by 2 group of microflora. One is called acidogens and there is methanogens. Purpose of acidogens is convert the organic material to organic acid, and purpose of methanogens is to convert the organic acid to methane and carbon dioxide. And I told you the acidogens grow much faster as compared to methanogens. So, size of the acidogenic reactor will be much smaller as compared to methanogenic reactor. So, we try to find out the amount, we find out, derive some kind of equation how to find out that volume of methane or volume of gas produced per kg of the per kg of the BUT removed or from the digester, that we can easily calculate. So, one problem we try to solve, and we show you that how this can be estimated. Thank you.